Okay, so we are looking at DNA replication. And our learning objectives will be for us to be able to cite evidence from Meselson and Stahl's experiment that enables scientists to differentiate between semiconservative mode of replication and alternative models. And then we are going to summarize how DNA replicates and identify unique features of the process of DNA replication. And then we are going to know the proteins that are involved in DNA replication. We're also going to explain the complexities that are involved in DNA replication that makes it to be bidirectional and also continuous in one strand and discontinuous in the other strand. And then we are going to understand leading and lagging strand synthesis. And then we'll discuss how enzymes proofread and repair errors in DNA that take place during DNA replication. And then we're also going to understand how DNA replication terminates, the termination of DNA replication. So the outline for our lesson will be, firstly, we're going to look at the 3 alternative models of DNA replication. And then we're going to look at the Meselson-Stahl experiment. And then we're going to look at the mechanisms of DNA replication. And then we're going to look at termination of DNA replication. So when we look at DNA replication, we're simply talking about the duplication of DNA, or in other words, DNA synthesis. So biologists at the time proposed 3 alternative hypotheses about how the old and new strands might interact during replication. So they came up with hypotheses that they thought that DNA could replicate. So the first one was conservative replication. So the first hypothesis of DNA replication is conservative, which is the one on the furthest left, conservative DNA replication. So under this hypothesis, they said that if the odd parental strands of DNA are separated, each could then be the conservative replication states that if the bases are temporarily turned outwards so that the complementary strands no longer face each other, they could serve as template for the synthesis of an entirely new double strand helix all at once. So this hypothesis is called conservative. Simply states that this would result in an intact parental molecule. Intact parental molecule. So you can see the parental molecule in blue there on top. And then according to the conservative hypothesis, it states that this would result in an intact parental molecule, which is the blue down there. So this will remain intact. And then a daughter DNA molecule consisting of entirely newly synthesized strand, which is that other color in orange. And then we have the semi-conservative alternative hypothesis, semi-conservative. So according to this one, if we have the odd parental strand of DNA, which is the blue there, each could then be used as a template. So each strand could be used as a template for the synthesis of a new daughter strand. Okay, so each of these strands could be used to synthesize a new daughter strand. So this hypothesis is called semi-conservative because each new daughter DNA molecule, would consist of one odd strand, the blue one there, and a new strand, that golden color there. This is the semi-conservative hypothesis. Then we have the dispensive hypothesis. So according to this one, if the parental double helix were cut wherever one strand crossed over another and DNA was synthesized in short segments by extending each of the cut parental strands to the next strand crossover, then there would be a mix of new, so you have a mix of new and odd segments along the replicated molecule. So you can see a mixture of the odd, which is blue, and the golden color there, the new. So you have a mixture. So this is a dispensive model of DNA replication. So these three models of DNA replications were evaluated. So Matthew Meselson and Franklin Stahl did an experiment to evaluate or to distinguish between these three models. So what they did is that they labeled DNA and then followed the labeled DNA through 2 rounds of DNA replication. So they grew E. coli, bacterium E. coli. So E. coli is small and grows quickly and is readily available in the laboratory. So it was a favored model of organism for the study. So they grew E. coli on a medium that contained 15N or heavy nitrogen in the form of ammonium chloride. So the molecule that was containing 15N or heavy nitrogen when they put it in the heavy nitrogen, it was grown, the E. coli grew for generations. And then they later transferred it into 14N or light nitrogen and observed what was happening. So they used density gradient centrifugation to separate molecules such as DNA, as in this case. When DNA is mixed with a solution containing calcium chloride and you do centrifugation at high speed, the solution forms a density gradient in the centrifuge tube, ranging from a region of low density at the top to one of highest density at the bottom. So you can have low density, which will be at the top, and heavy density at the bottom. So this is the experiment that they carried out to evaluate which of the three models of DNA replication was correct. So this was a prediction. What would happen if the replication was semi-conservative? What would happen in the generation 0 or in the heavy nitrogen? And if the replication was conservative or if the replication was dispensive replication. So after the experiment, what they verified was that DNA replication took the semi-conservative form of replication, which is the first diagram there among the predictions. It took the semi-conservative form of replication. So after generation 2, it showed that there was half low density DNA, which is 14N, and then half intermediate density DNA, which is a hybrid. Okay, so that was the Meselson Stahl experiment. So now when we look at the mechanism of DNA replication, the first one is we need to understand is that DNA replication is bidirectional. It means that it takes place in 2 directions as DNA replication proceeds. And it is semi-conservative. Okay. So bidirectional means that the replication begins at the site of origin and simultaneously moves out in both directions from that point. Therefore, replication bubbles grow in 2 directions as DNA replication proceeds. So you find that in prokaryotes, of which is going to be our concentration for our lesson. they only have one site of origin on each chromosome. Or when you look at eukaryotes, they have multiple sites of origin on each chromosome. Then DNA replication is semiconservative, which means that following replication, each daughter molecule of DNA will contain one intact parental strand and one newly synthesized strand joined by base pairs, as we have earlier looked at. And then we have replication fork, which is the Y-shaped region where the parental DNA double helix is split into two single strands and then is coupled. So these are the sites at which DNA synthesis is occurring. So we have specific set of proteins that are responsible for recognizing sites where replication begins and opening the double helix at those points. So we have proteins such as DNA proteins. So these proteins are activated by proteins that initiate the S phase during a cell cycle. An important point to note is that DNA replication and by extension, also DNA transcription begins at points rich in adenine thymine base pairs. So when you look at adenine and thymine, as we looked in the last class, we said that these are joined by double hydrogen bonds and not triple hydrogen bonds as in guanine and cytosine. So when you look at the double hydrogen bonds, it means that it is easy and it is with relative less energy to break the double hydrogen bonds of adenine and thymine base pairs than the triple hydrogen bonds of guanine and cytosine base pairs. And therefore, replication forks or initiation of replication begins at 80 base pair rich sites. And then we have enzymes called DNA polymerases, which are responsible for the synthesis of DNA. So when we look at DNA replication, it basically requires three basic things. For replication to take place, we'll need a template, which means that we need something to copy. Something to copy, which is the template. And then there's need of something to do the copying. In other words, the polymerase enzyme. And then we need the building blocks to make the copy, which are the nucleotides. So basically, in its simplicity, DNA replication will need a template, a nucleotide or nucleotides, and polymerase enzymes. So the polymerase enzymes are the one that catalyzes the synthesis of DNA. DNA polymerases can only copy a DNA template in the 3' to 5' direction and produce the new synthesized strand in the 5' to 3' direction. So we have the precursors, which are the Deoxyribonucleoside triphosphates, which are the nucleotides, which are the precursors for DNA replication. DNA polymerases cannot initiate DNA replication, and therefore it requires a primer. So primers are used during cellular DNA synthesis, which are simply short RNA strands and not DNA strands. So a primer is simply a strand, a few nucleotides long that is bonded to the template DNA strands and provides DNA polymerase with a free hydroxyl, 3' hydroxyl group that can combine with an incoming deoxyribonucleoside to form a phosphodiester bond. So when that primer is formed, it leads to provision of the hydroxyl group, 3' hydroxyl group, of which the polymerase then can use to synthesize or to add nucleotides to the chain. So DNA polymerase cannot initiate synthesis of new strands because one, it works only in the 5' to 3' direction. And it requires both a 3' end to extend from and a single-stranded DNA template. So RNA serves as the primer for DNA polymerase. So the primer is synthesized or catalyzed by the enzyme called primase. So this is a diagram which is simply showing an RNA primer. We have the template DNA there. Then we have the synthesis of an RNA primer by primase. We have this free OH group at the 3' end of the RNA primer, to which the DNA polymerase will then add the deoxyribonucleosides to the 3' hydroxyl there. And then the DNA parental or template strands are copied simultaneously at the replication forks, although they are going to run in opposite directions. Remember, during the last class, we said that DNA is antiparallel, which means that one strand will run in the 3' direction, and the other one, a complementary strand, will run in the opposite direction, which is the 5' to 3' direction. So this antiparallel nature of DNA, combined with the nature of the polymerase enzymes, will put a constraint on the replication process. Therefore, we find that DNA replication in one strand will be complex than in another strand. So the requirement of DNA polymerases for a primer means that on one strand, primers need to be added as the helix is opened up. So we have one strand that will need multiple primers, and then we have a strand that will only need a single primer. So the strand that is continuous, the strand that only needs a single primer is what is called the leading strand. Or the strand that is discontinuous, because it needs multiple primers, it's what is called the lagging strands. So on the lagging strands, there are DNA segments that are synthesized, and these are what we call the Okazaki fragments, which are simply named in honor of the man who first experimentally demonstrated this discontinuous synthesis. So here we need to note that on the leading strand, we only need one primer. On the lagging strand, we need multiple primers. And then the DNA fragments synthesized on the lagging strands are called Okazaki fragments. So the leading strand is formed by continuous copying of the parental strands that runs from 3' to 5' towards the replication fork. So the leading strand is synthesized towards the replication fork. Now that it is continuous, it leads into the replication fork and hence synthesized continuous. While the lagging strand is formed by discontinuous copying of the parental strand that runs from 3' to 5' away from the replication fork. So the lagging strand is synthesized away from the replication fork, or the strands that runs from 3' to 5'. So it's called lagging strand or discontinuous because it lags behind the synthesis occurring at the fork. So as the replication fork moves, it exposes gaps of single-stranded template of DNA. So what will then happen is that DNA polymerase 1 will move along in the 5' to 3' direction, removing those RNA primers ahead of it and replacing those ribonucleotides with appropriate deoxyribonucleosides. So once the RNA primer has been removed and replaced by DNA, then we have an enzyme called DNA ligase, which catalyzes the formation of a phosphodiester bond between the adjacent fragments. So this is the process of DNA replication. So we have the initiator proteins, DNA, a protein, recognizing the site for initiation of DNA replication. So you have initiation phase, elongation phase, and also the termination phase. We have the enzyme helicase. Have the enzyme helicase, which catalyzes the breaking of hydrogen bonds between base pairs to open up the double helix. So helicase catalyzes the breaking of hydrogen bonds between base pairs, which leads to the opening of the parental double-stranded DNA molecule. When that is done, there's the DNA molecule is destabilized. Remember, we said that the interior of the DNA molecule is hydrophobic. Therefore, when it is opened, the molecule becomes destabilized. Therefore, we need single-stranded binding proteins. These single-stranded binding proteins will thus stabilize the single-stranded DNA strand. So you have single-stranded binding proteins there and single-stranded binding proteins on the other strand, therefore stabilizing the two strands and preventing them from reforming back into a double-stranded DNA. And then we have topoisomerase enzyme there, which breaks and rejoins double.
Excuse me, sir.
Yes?
Excuse me, sir. Is it that the single-stranded binding protein stops the DNA from winding, or it winds it back on between the two?
Sorry, come again.
I'm saying the single-stranded proteins, is it that it prevents the DNA from winding, or it winds it back once it unwinds? Which one between the two?
The DNA, the single-stranded binding proteins, right? They stabilize the single strands. In short, they prevent the two strands from reforming. Is that clear?
Thank you. Yes,
clear?
thank you. Yes, sir, thank you.
Okay, welcome. Okay, so then we have topoisomerase. The topoisomerase there prevents excessive coiling during replication and then rejoins them in a more relaxed configuration. So topoisomerase prevents supercoiling of the DNA molecule. Okay, so we have I've talked about the helicase, single-stranded proteins, then we have the topoisomerase. And then we have the RNA primase, which synthesizes RNA primer. So we have the leading strand there needing only one RNA primer. And then we have the lagging strand, which will need multiple RNA primers. So the RNA primers are synthesized by primase, the enzyme primase, in that direction. Okay. And then we have the fidelity of replication, which is the ability to accurately replicate a template is very high. So DNA replication has a high fidelity. So it's accurate. So after the replication, you find that there's a process of proofreading that happens, which further increases the fidelity of the replication. You find that sometimes there could be an insertion of a wrong base. So for example, we learned that adenine should always bond with thymine and guanosine should always bond with a cytosine. But you find in certain situations where there's an error or an insertion of a wrong base. For example, adenine pairing with cytosine. So what will happen is that there's a process of proofreading. an exonuclease activity that is going to remove that base that has wrongly been bonded to the thymine. So the cytosine will be removed, and then thymine will be put in place. So you have the proofreading part. Then what happens during DNA termination in prokaryotes? So the completion of replication in prokaryotes occurs at corresponding points of the genome or the termination site, and is determined by two factors, which is meeting of the replication forks and also at points known as DNA replication terminus binding sites. So the meeting of forks occurs in the event that the DNA molecule has a closed circular shape, which is usually the characteristic of most prokaryotes or bacteria. So you find that you have circular. The shape of the bacterium is circular. Therefore, as a result of a continuous synthesis, the 3' and the 5' ends of each chain are joined. So when the two replication forks meet, then there could be termination. We're going to look at the events that takes place when those meet. Then we have structures called the catenanes, which are topologically linked circular molecules, which are formed. Therefore, DNA durase will then separate the rings or the circular daughter cells. The circular DNA molecule will be separated, leading to the completion of the replication process. And then we have the tear sites, which is a terminus utilization, which uses the terminus utilization substance, could also be involved. So the terminus utilization substance is simply a protein that binds to a terminus sequence and acts as a counter helicase. Remember, helicase does the unwinding of DNA. So you have the counter protein, which is the TAS. So when it comes in contact with an advancing helicase, it blocks further advance of the replication fork. The termination of DNA replication in eukaryotes is quite complex because one chromosome contains several points of replication initiation, and termination can occur in two cases, which is one, the collision of replication forks moving in opposite directions, and also in the cases where they reach the end of the chromosome. So now, the events that happens during termination of DNA replication, when two replication forks meet on the same strand of DNA, this is what is going to happen. We talked about convergence. So the forks converge until all intervening DNA is unknown. And then all the gaps need to be filled and ligated. And then we have the catenations, or rather decatenation, where the catenanes are removed by a type 2 topoisomerase. Remember, I talked about gyrase, which is a topoisomerase in prokaryotes. So there's decatenation. So the catenanes, those circular, topologically circular molecules of DNA, they are removed. And then the replication enzymes have to be unloaded. So those enzymes and the proteins have to be disassembled. Then DNA replication ends. And then of importance also is for you to know the enzymes, the proteins that are involved in DNA synthesis or in DNA replication, and be able to know their uses, what they do during DNA replication. So we've talked about what helicase does, what single-stranded DNA binding proteins do, what topoisomerase do, what primases do, what DNA polymerase does, and what DNA ligase does. So you need to know their functions during DNA replication. Do we have